All right, so hello everyone. Good morning, good afternoon, good evening from wherever you are joining us today. My name is Camille Pottinger and I serve as a health specialist here at NIA within the Division of Neuroscience. I'm joined by my branch chief of the Population Studies and Genetics branch, Dr. Damali Martin. I would like to invite you to the Minority Health and Health Disparities in Neuroscience webinar series. This series highlights opportunities for NIA grantees and other interested individuals to share research advances, lessons learned, and opportunities in the field. Before introducing the speakers for today, I would like to let you all know that at the end of the presentation today, we will allow time for questions. So please submit your questions and comments to the Q&A box, not the chat box, and Dr. Martin will ask on your behalf. Any questions that we do not have time to get to today, please feel free to reach out to the speakers directly. We will also share the Q&A box with the speakers, and they will also be able to follow up with you as well. Additionally, this webinar is being recorded, and we will send out to all of the attendees within the next week or so. So let's get started. It gives me great pleasure to introduce this next panel of speakers for the series, Dr. Sid O'Brien, Dr. Alexandra Clark, Dr. Raul Ventimilla, and soon to be Dr. Mohammed Husseini. The title of their presentation is Risk for Alzheimer's Disease Across Diverse Populations, Lessons Learned, Opportunities to Advance Science, and Recent Findings in the HABS-HD study. Dr. Sid O'Brien is the PI of HABS-HD, which is the most comprehensive study of Alzheimer's disease among the 3 largest racial ethnic groups in the US, including African Americans, Hispanics, and non-Hispanic whites. The goal of HABS-HD is to understand the life course factors, including biological, sociocultural, environmental, and behavioral, that impact risk of Alzheimer's disease in late life. This work will ultimately lead to population-specific precision medicine approaches to treating and preventing Alzheimer's disease. In addition to being a global leader in health disparities and cognitive aging, Dr. O'Brien is also a global expert in the use of blood-based biomarkers for the generation of precision medicine approach to novel diagnostic and therapeutic strategies for Alzheimer's disease, Parkinson's disease, dementia with Lewy bodies, and Alzheimer's disease among adults with Down syndrome. Next, we will hear from Dr. Alexandra Clark, who is an assistant professor of psychology at the University of Texas at Austin. She is also a clinical neuropsychologist and an inaugural early career faculty fellow for the HABS-HD project. Dr. Clark will highlight her recent work identifying distinct psychosocial behavioral phenotypes in HABS-HD and discuss evidence that these phenotypes displayed varied cognitive and AD biomarker outcomes. This work illustrates how data generated by HABS-HD can be used to enhance our understanding of important patterns of risk and resiliency to AD in racially and ethnically minoritized older adults. Next, we will hear from Dr. Raul Ventimilla, who is an assistant professor at the University of North Texas Health Science Center and a researcher with the HABS-HD study. As a previous health disparities in Alzheimer's disease and mild cognitive impairment NIA diversity supplement recipient, Dr. Ventimilla will highlight his recent work on cardiovascular disease impact among Hispanics living in the US. Specifically, the relationship of MRI markers of small vessel disease and cognition in Mexican-American cohort. Data from the HABS-HD cohort can be used to advance our understanding of the effect of cardiovascular risk factors in Hispanics. And last but not least, we will hear from Mr. Mohammed Husseini, who is a DO PhD candidate at the University of North Texas Health Science Center, Texas College of Osteopathic Medicine, also a previous NIH T32 Neurobiology of Aging Fellow and a current Alzheimer's Disease Neuroimaging Initiative Health Equity Scholar Program Fellow. Mohammed will highlight his recent work on genomics and health disparities among minorities in HABS-HD and elaborate on the need to further investigate AD using ethnicity-specific methods in order to accurately capture disease risk in each ethnicity. This research is imperative to bridging the health disparities gap in AD, so that healthcare providers can utilize tailored approaches to patients of different ancestry, achieving better outcomes. And so with that, I will hand it over to our first speaker for today, Dr. Sid O'Brien. Thank you very much.
Thanks for having me, Camille. So I'll kick this off. First, I just wanted to say thank you. Thank you to Camille and Damali for having us here today. And thank you to Ali, Raul, and Mohammed for being part of this. This is my first time that I've actually been able to introduce this study and then watch the next generation of people to run it actually speak. So it's a huge honor for me. And thanks to y'all for being here. This project and this data is yours. It's not mine. It belongs to the scientific community and to the communities which we serve. So we want you to use it, and we're going to talk about what is here, how they will discuss how they've leveraged it, and in hopes that you will do the same. So we're going to talk about risk factors for Alzheimer's among diverse populations, opportunities, et cetera, in HABS-HD. Next slide, please. So I begin with the thank you. This is the local Institute for Translational Research. This is not the full HABS-HD team. I couldn't fit that on a slide. These are the local folks who actually see participants, conduct the scans, et cetera. So just thank you to the team because they're absolutely amazing. And for those of you who want to come to the annual meeting, you get to meet them. Next slide, please. So why are we doing what we're doing? So in my opinion, you cannot study the biology of Alzheimer's disease in the absence of the environment and sociocultural factors, nor can you study sociocultural environmental factors influence on brain aging in the absence of biology. And so what we did here is something that hasn't been done to date at this scale, which is really an honor to be part of, an honor to lead. And so this is the graphic that describes the U19 projects. And at some point, if y'all want to trainees would like to learn about U19s, I'm happy to discuss that. Basically, what you'll see is on the top, project 1 is studying the ATN framework among diverse communities. And we're studying the biology of amyloid, for example, looking at not only imaging, but also looking at genomics, looking at proteomics, exosomes, et cetera. Same with tau and neurodegeneration. For those of you who enjoy the analytics, what you'll see is this is basically a structural equation model. So then we have been looking at an ATN VMI framework for a very long time. So we were excited to see the new 2024 framework, including VNI. And so project 2 is expanding, looking at these vascular, metabolic, and inflammatory factors that we and others know are associated with Alzheimer's, including the basic biology of Alzheimer's, but also these factors vary by race and ethnicity. Then what we do is right through the middle, which is project 3, you'll see is all of this is being examined through a health disparities framework. So we can understand how sociocultural, environmental, and behavioral factors impact, moderate, mediate the expression of these things, because I personally believe that a precision-based medicine approach to risk reduction will also include intervening even at population levels. Next slide, please. So HABS-HD, where are we so far? We've already enrolled over 1,500 Hispanics. It says Mexican American. I need to fix my slides. We began enrolling Mexican Americans because that is the largest Hispanic group in the United States, especially in Texas. However, demographic trends have been changing, and our community has been asking for years why. And so we actually changed our recruitment. So anyone self-reporting of Hispanic now comes into the study, and we've enrolled several hundred non-Mexican Hispanics. We've also enrolled over 1,200 African Americans, which I'm super proud of because we launched enrollment of the African American community post-COVID, right in the middle of COVID. And so demonstrating how effective the group has been, over 1,400 non-Hispanic whites. We actually have over 1,000 people on a wait list for the study. So I can talk about the community engaged approach at some point. 30 and above, we added, so at the U19 phase, which is really an expansion and a renewal, we added 1,500 in the 30 to 49 group. So that's the newer group that we're enrolling. We always replace for attrition. And so the cohort will never dwindle down, which is why we have more than 1,000 per group above 50. We've been doing MRIs since long time, really. We added amyloid, and then we added both amyloid and tau. The database already contains over 5,000 MRIs. We're pushing 4,000 PET amyloid scans, and before the end of the year, we will surpass 3,000 PET tau scans. So in terms of imaging data, it is the largest repository of brain scans among diverse communities, because 66% of participants are non-white. We do functional exams, clinical, sociocultural. We have a biorepository over 500,000 aliquots. We are in the process of doing full systems biology. We have already deposited whole genome sequencing from 1,500 people, with another 1,500 going into NIGADs before the end of the year. Whole genome sequencing as well as GWAS is available. We're doing target and untarget proteomics, metabolomics, transcriptomics, you name it. So think basically community-based multi-ethnic ADNI. That's how robust this study is. And all of the data is freely available. And I'll get into that a little more. Next slide, please. And so one of the things that when we started, for those of you who would like to and plan to get in recruitment of underserved and diverse and marginalized communities, I've been doing this a long time. And I tell people that I've built a career on doing things that I was told I couldn't do. And so we were told, well, can't recruit rural, can't recruit Hispanic, can't recruit Black. Then we did. So far, our team has recruited over 12,000 people from single site studies, all from underserved communities. Then it was, well, you can't recruit in imaging studies, then you can't recruit to PET scans and blah, blah, blah. So, but what you see here is our scan rate by ethnicity is exactly where we would want it to be. And our goal's always been 60-40 male to female. And so what you'll see from these graphs is that, the team has done absolutely wonderful. This data is freely available, and we are more representative than others anticipated. Next slide, please. And so what are some lessons learned in core tenets? How has this group been so successful? And one of the things that I think that people skip is tracking where the mistakes are. As you're growing a research program, you learn, right? Failures are actually the best teachers that you're ever going to have. And I have failed more than most. And so what I believe is very important is to learn from these things. And what our team is built around are these fundamental tenets, right? So transparency and communication. We have weekly meetings with the team that are operations. These aren't science meetings. These are, we go in, what do the participants need? What do the community needs? Every time we build something, we build it to where it needs to be in the future, not to what we need now. And so if anyone has something, they say, well, we'd like to do this. And I was on a workshop and even a dissertation the other day, beautiful, elegant design, not scalable. And so from my standpoint, to meet the communities I am really charged with and honored to serve, we have to have ways of scaling to those things. And so anything we do, from an electronic data capture system, a scheduling system, neuropsych testing, imaging, everything has always been built to, you want to see 100 people, we'll build it so you can see 500. You start getting to 300, okay, now how can I scale it to 1,000? And so you always want to be thinking about what you're doing in terms of, can it meet the needs of where you want to be, not where you are. And then the next part that feeds into that is that rapid monitoring. I actually get a weekly dashboard that many have seen, that I see what's going on in the study. I know how many phone calls were made, how many people were scheduled, how many people were shown up, how many came to this scanner, and it's all there. That's not for micromanagement purposes, because I don't have anything to do with the day-to-day aspects like that. I'm looking at big picture so that we can say, for example, we have a backlog and consensus review, so that, all right, maybe in June, we're actually going to slow down the number of participants we're seeing because we're already going to meet our metrics and milestones. So why don't we slow down a little bit, give the team a little bit of a break, make sure they're taking their vacations, and then get caught up on consensus review. So you can find that balance, but also you're not trying to move the Titanic. You can actually adapt real time, which leads to that adaptability and flexibility. I'm not a fan of doing things because that's the way you've always done it. Challenge the status quo. That to me is what's important. It's about helping patients. It's about helping the community. And the only thing that matters to me is, am I helping the community? Many people know why I do this. It's personal for me. And so if I'm going to help those patients, I'm going to help loved ones, I have to realize that I don't know everything and I'm oftentimes wrong. And so learn from that so that you can be adaptable, flexible, and you can then meet the needs. The other piece that I feel gets looked at in the wrong manner is quality control. Most people think of quality control when they get to the database. That's really not the best approach. We build in quality control aspects into every item of the electronic data capture system. So there's a minimum and maximum. We can actually even look at the trends in its individual tests, et cetera. And so we build a quality control at the data capture level so that we're preventing having to fix things later on. But then when we find things, we fix them and move forward. And the last piece is a little bit more of a bigger picture sort of thing, but it gets to what I said earlier around the monitoring and flexibility is I always know where we are. The team knows where we are. And then we're predicting out so that right now we already know exactly or estimated where we will be at the end of this year. We also are building a system so that by the time we start year 3 of the U19, we will know our predicted year 3 total numbers of everything, year 4 and year 5. So based on all of these components, we can predict because we know no-show rates by month. We know how many people are scheduled a week. All of these things allow you to have a better sense of actually what's going on. And this allows you to meet the needs of your community, but also the needs of your team. You don't do anything. Your team does wonderful things. We all are part of something bigger. And in my opinion, no person in human society has ever done anything to actually impact humankind alone. Everyone has worked part of a team. And so this is what this system is about. Next slide, please. And so one of the things that I wanted to bring up is some of the core philosophical tenets about approaching a community. I'm blessed to actually help numerous large-scale programs in building community outreach and engagement to enroll diverse communities is something that I'm obviously very passionate about. There's some core fundamental things, though, I believe we need to remember. And the one that I don't have on here that I've got to get in there because I say it all the time is just get out of your ivory tower. If we sit in our ivory towers and wait for people to come to us, we will never change the status quo. There's tons of literature showing that individuals from marginalized communities don't show up to specialty clinics, and it's not their job. In my opinion, health disparities are the fault of the scientific and medical community, so it's our job to fix it, and we do so by getting into the community. So you create partnerships. I've seen so many people go to community events and they're trying to build something and they come off like, I'm trying to save you. That is the fastest way to really, really offend people. The communities know what they need far more than you do. Go listen, go learn, go ask questions. Meet the participants in the community where there are. That's one of the beautiful things about what we've been able to do is, like, for example, we have a state-of-the-art imaging center. There's never been a patient in it. There will never be a patient in it. It's designed as a community entity. And so it's very nice. It's very warm. It's very friendly, et cetera. And so our team goes all through the community. And then people, when they come here, they already know us. One of the things that I think is important for people to realize, I've built clinics too. Building a clinic takes time. You build community trust, et cetera. Well, this is no more difficult. It's different and it's a lot more fun. Giving back. This is something that I argue with people all the time. Well, we can't give this, we can't give that. And my response is, it's not yours. It's not your data, it's the community's data. The community wants it, figure it out. If the community needs feedback, well, that's your job, figure it out. And so we give back tons of information. We're even evolving what we give back, expanding what we give back. And that, I believe, is one of the single biggest reasons people stay in our studies, is the give back and the community ties. When you're thinking about the community, I always think about, I phrase it as buckets. And so there's different groups. You don't just go out. You need to understand who's your target audience, who are your stakeholders, who are you engaging? Community organizations, elected officials, religious organizations, medical, then your participants themselves. And so you can build strategies that I'm happy to help how to do those things. But this is more of a philosophical sort of, how do you approach the community from my experience. Next slide, please. So what about educational and training opportunities? So Dr. Clark, who's going to speak, is actually the first faculty member of the HABS-HD training, scholars training program. And so you'll see here, we do this every year. We've actually doubled the size now. And so we're going to be recruiting even more into the cohort beginning in year 3. I think the application cycle just ended. So if you're interested, I suggest you look there, because apply it in cycle 4. The scholars have been amazing, and it's just such a wonderful opportunity that you get to meet and learn from some of the brightest people in the world who happen to be good personal friends of mine. Next slide, please. This is really just a snapshot. And one of the things that I like for people to realize, and when you look at this, you don't see the overwhelming names of these grants as PIs of HD. So Christine Yappy is a MPI, and I'm on that grant, and that was the first ancillary sleep study grant. However, Boone Lee-T, Zachary Green, Rachel Buckley, Karen Meeker. Karen Meeker is a junior faculty member, got a KO1. And so, Yang Gang Shi is part of our project at Lonnie. The Dwight German, for example, is at UT Southwestern. So the distribution of the grants have not been to PIs of this study. We encourage you to go get grants. We want you to leverage the data. And the other thing that I think is important for people to realize and remember is there are tons of opportunities for you to go after and secure grants, leveraging data and repositories. You do not always have to create a new cohort. That's what this is for. It's your cohort. Leverage it. Next slide, please. So the data is readily available. There's the website for it. All item level data is there. Raw imaging files are data there. Getting access to the data is very simple. Fill out a proposal. If you're a trainee, make sure you have a mentor on it. And then normally basically within a few days, you're going to have your data. So as soon as I personally go in and review most of these myself, and as soon as I click approve, within a few hours, you have a link to tell you go download your database. The data will transition to Lonnie, and it's in the process of that, which will make, I think, access even easier. In the last 24 months, we've given out over 350 databases. I think we have now covered 12 or 13 countries. And so all the way, we've even had high school students download data and use it. And so very proud of the teams for that. Next slide, please. And I'm going to stop there for the team. Next, Dr. Clark. You're on mute.
Thanks, Sid. I have the privilege of talking a little bit about why we need to think about the lived experiences of the community members that we're studying, especially if we really want to understand why we see differences in Alzheimer's disease and the manifestation of the biology of the disease. Okay, next slide, please. So I want to start with acknowledging that we're here and we're all very passionate about studying these racial ethnic disparities in AD. And they're avoidable and unjust, and we can do something about them. They're here largely because these racial ethnic groups are more likely to be exposed to risk factors and less likely to be exposed to health promoting resources. Okay, these inequitable resource distribution patterns have created these disparities that we need to do something about. Next slide, please. When we think about these health depleting risk factors and health promoting resources that we want to capitalize on, it's important for us to recognize that the NIA has put together this beautiful conceptual research framework for us to leverage in our research initiatives, okay? And we can largely categorize these risk factors and resources into different levels of analysis that are kind of at the top of this chart here, okay? We've got environmental risk factors and resources, sociocultural risk factors and resources, behavioral risk factors and resources, as well as biological. And these environmental, sociocultural, and behavioral risk factors are really kind of one of the reasons why we see changes in the biology of AD. Okay. And it's important for us to think about, not only do we need to study these individual risk factors and resources listed in this framework here, but understand that people are exposed to an array of these across the life course. And we need to really kind of do a better job of taking into account and characterizing that exposure, especially if we want to understand these late life disparities in AD. Next slide, please. So, when you look at that chart, you probably think of some research studies, and in part, we know that if we focus on one of those individual boxes, okay, we're like SES, we know the effects of SES on cognition, we know the effects of stress on cognition, and we've done a really good job of studying individual risk factors and resources, okay? But what I want to encourage us to think about is to move beyond those individual level characterizations and paint a better picture of the people that we're studying by taking into account exposure across the life course, across multiple levels of analysis, okay? And really, this is kind of in line with precision health perspectives. We need to understand that there are different experiences that people have. Those different experiences impact our health and our late life outcomes. And we need to do a better job of characterizing those experiences if we want to understand some of the outcomes that we study. Next slide, please. We've started to do some of this in HABS. HABS is such a rich data source, and the team is fantastic. I have nothing but fantastic things to say about them. And I want to kind of take a look at this framework and say, instead of just studying one of those circles, we've started to use some modeling methods that allow us to characterize multiple risk and resiliency factors, as highlighted here. And we're going to think about these risk factors, especially when we start to understand the biology of Alzheimer's disease. Okay, so let's move beyond just individual levels of analysis and characterization to really multi-level characterizations of all these variables that we know play a key role. Next slide, please. So we've started to do some latent profile analysis and some characterizations across multiple levels of resources and psychological variables that we think are important and play a role in the manifestation of AD, okay? And we've used LPA and modeled some important variables related to income, occupational complexity, and psychological functioning in the data set to realize that there's some unique variability in the data set and groups of people that we can identify that are likely important for us to think about when understanding who's at risk, who's at increased risk, and who may even be resilient to AD, okay? So we've used LPA or cluster analyses actually to identify that there are three groups of people in this data set. There appears to be a low resource, high distress group. So people that have low levels of economic resources and social support, high levels of psychiatric distress. There's a high resource, low distress group. So people that have more access to resources and low levels of psychiatric distress. And then people that don't have a lot of resources or low resources and low levels of distress, okay? And one of the things we've been really interested in is now that we've characterized that there are groups of people with varied experiences in this data set, well, what does that mean? Next slide, please. Is this something that's important for us to take into account when it comes to understanding Alzheimer's disease risk? Is Alzheimer's disease risk the same across all these distinct groups of people that we've identified? Next slide, please. So, not only identifying these groups of people, we've started to ask, all right, well, what do they look like in terms of subjective and objective markers of cognition? And do they differ also on Alzheimer's disease biomarkers? And what we've come to appreciate is, if you take a look at this slide here, that this low resource, high distress group has significantly worse memory or more subjective memory concerns compared to the high resource, low distress and low resource, low distress group, okay? So they seem to be an at-risk group, or a group that really is concerned about their subjective memory experiences. And interestingly, we don't see too much of a difference between those high resource, low distress groups and low resource, low distress group. Next slide, please. If we take a look at objective markers of cognition too, and move beyond just self-report measures, we see similar kind of changes, okay? We see, it looks like my figures got a little messed up here, but I'll say that we saw a similar pattern that we saw on that previous slide, that high resource or low resource, low distress group had worse cognition when compared to those other two groups, okay? So there are, not only do we see that they have higher memory concerns or complaints, but they also, this is translating into objective markers of cognitive concern and impairment as well. Next slide, please. Then, we want to not just think about these lived experiences, but also kind of understand the biology of Alzheimer's disease and risk across these groups as well. And we see that, again, there's some significant differences between each of the groups on plasma NfL markers, okay? The low resource, high distress group had significantly higher levels of plasma NfL compared to the high resource, low distress group and the low resource, low distress group, okay? So there's something really vulnerable about this low resource, high distress group. But we also want to recognize that there's some seemingly resilient groups in the data set that need to be studied, and that we can really capitalize on understanding these people, especially as we think about prevention and intervention initiatives that are relevant to racially and ethnically minoritized older adults. Next slide, please. So I just want to kind of summarize briefly that we can use multi-level phenotyping or characterization methods to allow for a more nuanced understanding of risk and resiliency to Alzheimer's disease in the HABS data set. And again, let's move beyond those single level characterizations to taking into account multiple variables so that we can have a good understanding of people's true kind of lived experiences, which are quite complex. We saw that low resource, high distress group was an at-risk group, okay? They had poor executive functioning, more subjective memory complaints, and higher levels of plasma NfL. So if we're going to direct our resources and really kind of understand and identify groups of people that are at increased risk, these are the people that we want to focus on. And, we oftentimes focus on risk and we talk a lot about risk identification, but I want to really encourage us to also think about resiliency in the HABS data set, okay? We saw a group that even in the face of lower levels of resources, that low resource, low distress group, they were doing pretty well from a psychiatric standpoint and were resilient. They seem to be similar to their high resource, low distress peers, okay? So let's not just think about risk, but let's also think about resiliency so that we can capitalize on that. I just want to highlight that we're doing some really fun studies to kind of better understand these groups moving forward. We're looking at biological aging across these groups, longitudinal neuroimaging outcomes, and trying to see, all right, who progresses to MCI and AD at different rates, okay? So thanks, everyone. Next slide, please, for your time and attention. I'm happy to talk about that research study in detail in the future, and thanks for listening.
Hello, and thanks for having me here. I'm gonna speak about a preliminary study we have, and that's about a predicted value and weight of factors associated with cognitive performance in Hispanics from the HABS-HD cohort. Next slide, please. We all, of course, know that there are multiple risk factors that have been associated with cognitive performance, cognitive decline, and dementias. But unfortunately, the efforts up to date to identify these risk factors relied on meta-analysis. And we all know that the problem with meta-analysis is that they are vulnerable to bias, basically because the different methodological in the different methodology in studies, right? We have different quality of the samples, for example. We have different inclusion-exclusion criteria. Each study have a different way to measure the predictors and many others. So that's not really the best way to identify risk within the same sample. Now, also, even if we talk about meta-analysis, there is a lacking of this analysis in Hispanic populations. So for that reason, we thought that's very important to do an empirical initial analysis of the risk factors associated with cognitive performance and within our sample. And with that, next slide, please. We decide to choose 36 risk factors in different cognitive domains and see the relative importance of these factors using random forest survival analysis. Next slide, please. Based in the prior experience and prior research within our cohort, and also using, of course, what is published, the Alzheimer's Association conclusions, the Lancet Commission of Dementia conclusions, we use demographic, the obvious demographic factors like age, education, and we added ADI, the Area Deprivation Index, health factors like the diagnosis of the major cardiovascular risk factors, and also continuous measures. We have a few psychosocial measures of stress, social support, worry scales, depression scales, subjective complaints, and biomarkers, blood biomarkers associated with AD like tau, A-beta 40 and 42, inflammatory biomarkers, and we use white matter hyperintensity as a biomarker of small vessel disease, brain small vessel disease. And we have a few neuropsychological tests like MMSE, SEDLT, Trails A and B, FAS, animals, digit symbol substitution, among others, to measure different cognitive domains. Next slide, please. Here we have part of our participant characteristics. A few things that I want to say first to understand our cohort is, first, we do have, in this sample, the majority of the cohort is Mexican American. We just have, out of the 1,200 or 1,300 participants, around 100 non-Mexican Latinos from like 7 or 8 different countries. So really the cohort can be considered Mexican American at this point in this analysis. Also, we have a mean age of 60, but really, again, 1,200 participants in this sample were 50 and above. We just have 100 participants that are 30 to 50. So another thing that's very important really to understand the cohort is that look at that 70%, the mean of 70 of the ADI, the Area Deprivation Index rank goes from 0 to 100, and the higher the value, that means the most deprived neighborhoods. So really in 60% of our Mexican American cohort live in the most deprived neighborhoods. And that's something very important to take in consideration. And the right, you can see the random forest model accuracy. And we can see that all in every single domain, the R2 are higher than 0.8. So really, this model shows a very good accuracy. Next slide, please. Before we get into the results, let me do actually two disclosures. The first one is that you can see almost in the bottom of the importance scores, this variable that says GD, total tau, CRP, I have to apologize. That variable does not exist. That was created by mistake when I was creating the database. And when I realized that we have that variable, it was too late to change it. So that's not a real variable. But I told with the biostatistician after I found that, and we don't think that's going to make a difference in the results. And another thing to take into account here, please, is with the GFAP. Out of the 1,300 participants, less than 20% really, closer to 10% have GFAP values. So really, these GFAP results needs to take with very, very cautiously because it's not really reflected. But again, we don't think the rest of the results is going to change because of that. The first thing to notice here is really interesting that for each cognitive domain, if we go over the first 10 in relative importance, the first 10 factors, they are different for each domain. And of course, some of those like income, age, gender may repeat, but the order they are completely different depending on the domains. And another very interesting fact we found is that in the previous slide, you saw the prevalence of the main 4 cardiovascular risk factors in our cohort. And we were expecting that those factors, the categorical factor of diagnosis of hypertension, diabetes, obesity, dyslipidemia, were going to be important factors, at least in the 10 top. For our surprise, in every single domain, the categorical factors are in the bottom of the list. But when we use continuous variables for health, like BMI, systolic blood pressure, diastolic blood pressure, A1C, total cholesterol, of course, they are part of they go up in the relative importance score. Next slide, please. The same thing we can see here in attention, executive function, and processing speed. Please do not pay attention to the GFAP, but you can see here white matter hyperintensities shows in this part with biomarkers. And if you see here between the first 10 in each one of the domains, biomarkers are always there. Even in higher levels than the clinical risk factors. Next slide, please. And finally, we have here the language, phonemic, and semantic fluency. And you can see again, A beta 40, A beta 42, CRP, VCOM, the biomarkers are in the top. And white matter hyperintensity is present here also. Next slide, please. So really, when we see and compare the 10 main risk factors in each one of the domains and choose the 5 that are present in the majority of domains, we really find that biomarkers, especially AB40, AB42, CRP are in the top. Actually, AB40, 42, and CRP repeat in 7 of the cognitive domains. White matter hyperintensities, the MRI marker we use repeat in 6 domains as one of the top 10 factors. And also, like I said, again, the categorical health factors were in the bottom of every single domain. And that's not really, looking at the literature, it's not really that surprising because literature is saying now that it's way better to use continuous variables like BMI, systolic blood pressure, cholesterol, abdominal circumference, instead of categorical variables. So that might be one of the explanations. Of course, this is just a preliminary analysis. We are, like Dr. O'Brien said, recruiting more and more participants. So we need to do a further analysis in a bigger sample to increase, of course, the prediction accuracy. We need to include novel biomarkers, maybe NFL. We need to do the AB42, AB40 ratio. I want to add more than white matter hyperintensities, also maybe volumes related with AD areas, signature areas like hippocampal volumes, for example, and other measures of small vessel disease like microhemorrhagia, microinfarcts that it needs to be added to the risk factor models. More health habits that are in the literature like alcohol intake, drug use, that I don't have it here. In socio-demographic factors, it will be very important to have an historical ADI, because the one we use now is just a cross-sectional and based in where the participants are living right now at the time of visit. We want to do an historical ADI so to see how it affects cognition, and of course, another neuropsychiatric indicators. And after we have the main risk factors in our cohort, we think it's very important to do a longitudinal analysis of factors and see really the factors related with cognitive performance, which ones are actually related to cognitive decline and can predict MCI or dementia. Next slide, please. I just want to thank you, all the researchers that are here in the ITR HABS-HD, all the project managers, coordinators, all the personnel, and most of all, I want to thank the HABS-HD participants and their families for trusting us, for being us through the years. Thank you.
Hello, everyone. Thank you for having me here today. It is my honor to present to you some of our recent findings in the HABS-HD genomics core. Next slide, please. So I'm going to jump right into it. One paper that I recently published is about the genotype frequency differences between the Mexican Americans and the non-Hispanic whites in the HABS-HD study. When I first started my PhD with Dr. Barber, we were trying to go through the literature and for me to get a better understanding of the current situation in the field, because my background is clinical medicine and neuroscience. So genetics was new for me. But what I was realizing is that the vast majority of the literature is on people of European ancestry for Alzheimer's. And having access to a diverse data set, we were interested in looking at the top 10 Alzheimer's risk alleles that we pulled from Kunkel et al. 2019 genome-wide association scan. And we chose the top 10 because these have the highest effect size. So we wanted to compare how frequently, what are the genotype frequency differences in between the two populations. And you can see that these genes are responsible for a variety of important biological functions. So in table 2 at the bottom, you can see the cohort that we ran our analysis on. We had roughly comparable amounts of participants between the two groups. And the only noticeable differences between some of the values here, you can see that education years, there's a big difference, and diabetes is almost double in Mexican Americans. Next slide, please. So this is a lot of data, but it's actually very, very simple. The first thing that we did was look at the ApoE4 allele positivity between the two groups. And ApoE is perhaps the most famous of all of the Alzheimer's risk alleles. So that was the first thing that we wanted to see is what was its positivity between the two groups. And interestingly, we had 30.31% positivity in the non-Hispanic white group and only 18.41% in the Mexican Americans. And then on the left panel, ApoE has 2 SNPs that we looked at, and then the other 9 are in the right panel. So total is the top 10. And everything except the ones in a box on the bottom, TREM2 and CD2AP, had statistically significant differences between the two genotypes, between the two populations in their genotypes. And you can see on any of these figures, let's see, for ABCA7, there's 3 possible genotypes that you could have. You could be homozygous CC, heterozygous CG, or homozygous GG. And that was essentially the analysis that we did. We wanted to see at what frequencies were these base pairs for each of the alleles between the two populations. Next slide, please. The other question that we had is what kind of genetic architectural differences were there? And this is data for linkage disequilibrium. Linkage disequilibrium is essentially the non-random chance of blocks of genes being inherited together. During DNA replication and recombination, genes are shuffled and inherited in essentially random patterns, but linkage disequilibrium describes the non-random chance of genes being transmitted or inherited together. So the red color, the more red color is a higher linkage disequilibrium, indicating that these genes and their neighbors are inherited in blocks together at a higher frequency. And as you can see, the linkage disequilibrium values between non-Hispanic whites and Mexican Americans for these genes are quite different, indicating that there are genetic architectural changes, differences between the two groups. Next slide, please. So our analysis shows that the top 10 risk alleles for Alzheimer's and the single nucleotide polymorphisms there are quite different. And you can see on the right-hand side, these top 10 genes, they fall into buckets of things like cholesterol shuttling, immune regulation, cellular morphogenesis, or cell signaling. And this is interesting because when we look at changes or differences between different populations, you have to start thinking about what are some of the more downstream effects and manifestations of comorbidities, other things like epigenetic changes in environmental factors that can eventually affect the comorbidities that these people have. So further research is definitely necessary to elaborate the differential health outcomes that are experienced by minorities. And this helps kind of segue into my next work. Next slide, please. I'm interested in exploring a polygenic risk score for Alzheimer's disease in our populations. So what is a polygenic risk score? Essentially, it is an algorithm to stratify risk, and using your genetics, we can come up with a score, a value of high risk or low risk or intermediate risk for developing the disease or the trait, in this case, Alzheimer's. And I came across, I was motivated to do this because I came across the MyGenes clinical trial, which was a cardiovascular disease polygenic risk score study. Traditionally, for cardiovascular disease, the Framingham risk scores and the American Heart Association risk scores, they take into account traditional things like blood pressure, cholesterol, smoking, and these are used to diagnose and initiate treatment, which is to lower the LDL with statins. However, in the MyGenes study, they discovered that adding a polygenic risk score to the criterion increased the predictive power, and treatment benefit was highest to those who had the highest polygenic risk score, and that most importantly, disclosing the status, the polygenic risk score status to the patients at the time of treatment initiation had allowed for better outcomes and better patient compliance. Next slide, please. So this is some very new data that I recently produced. It's a pilot. I am still learning how to do this, and part of my training was to use a small subset of Mexican Americans from the HABS-HD study, where we took 665 individuals and constructed a polygenic risk score. And on the left, you can see that the p-values thresholds, there's a variety of them, but the ones that reached the best fit, in our case, 0.015, this indicates that this is a good direction for the pilot study. So it's having it's reaching significance. And we constructed two models. The first model was taking only into account the ApoE4 status, and that yielded an area under the curve of 0.54, which is only a little bit better than chance, but that's what we would expect for this one. And then we did the study again, but accounting for age, sex, ApoE status, and the first 5 principal components. And the area under the curve improved to 0.605. And this number may seem kind of low, but in fact, it's quite good for a starting point using just these covariates and in a small sample size. And my hope is to continue this work with a larger group of these individuals and to fine tune some of the variables and factors and all the statistics. It's something that I'm learning how to do right now, actually. Next slide, please. So the future direction that I want to go into, as I said, is to leverage the entire HABS-HD cohort and all ongoing data collection. And after I've constructed these scores for HABS-HD, I'm interested in incorporating data from other large data sources like ADNI and TARC. We think that it would be very interesting to do some comparisons across different populations and further fine-tune the model for better accuracy. The other thing that I'm very excited about is to use, rather than just a binary trait outcome for the polygenic risk scores, such as Alzheimer's or no Alzheimer's, I'm curious to try a continuous variable using the imaging. I think that would give us a little bit more insight and perhaps better accuracy for the model. And then the second direction that I want to go to is to try and construct a methylation risk score. So we have all the epigenetic data, and while this has not been done for Alzheimer's in minorities, there has been a methylation risk score that's been done for major depressive disorder. And I'm hoping that we are able to use from their methods and their model a similar way to construct some sort of epigenetic risk stratification, and hopefully that will have significant results. Next slide, please. And I want to thank all the people that have been involved and helped, my committee members, National Institute of Health, the funding, and CIPRIT, as well as all the people at the HABS-HD study team, and Dr. Giuseppe Tosto, who trained me in some of these methods. I've learned a lot from his lab, and I really want to thank everybody involved. Thank you.
Great, that was fantastic. Thank you so much to the entire team. One of the reasons why I asked Sid and his team to present at our webinar series today is that I truly do believe that this is a model for how you run a study, how you leverage a large investment that NIA has made into a study in terms of not just sharing the data, And I'm quite sure people are not used to hearing my data is free, but it really is free. So not just in terms of sharing the data, but also in terms of training a diverse group of next generation researchers. And so, you know, as many times as I've heard you guys present, I'm always amazed when I see you again. So thank you so much for joining us today. And I did notice that, you know, you guys were trying to answer a lot of questions as they were coming in, which is great. But they're still popping in really quickly. So we're just going to go straight to the questions. I'm going to start with the ones that you haven't responded to as yet. And if we have time, we'll go to the ones that you've answered. And if we don't have time to get to your question, I really apologize. But I guarantee you that if I send your question to this group, they will respond to you. So we'll do that also. So the first question is from Arubella Reddy, and this is to you, Sid. There are new groups coming up that may be at risk groups right now. Do you identify and how do you support the outreach?
Yeah, I mean, our outreach goes across, I froze for a second. Our outreach goes across the community. For recruitment into the study, we only recruit the 3 groups that we're currently capable of going recruitment. We are looking at ways of expanding because we have other groups that are asking, hey, we'd like to be part of the study as well. When we're in the community, we will speak to anyone. So we will do outreach and education to any community that would like. I agree with that. And, you know, my goal at some point, and Damali knows this, is I want to see this study go completely national and global so that we can just continue to expand and reach communities across the entire country, because there's so many ways that we can reach communities that are underserved that we currently don't serve. And that's very important. Right now, we're not recruiting into the study because we don't have the capacity to do so, but our outreach aspects, absolutely, we'll go speak to any group who wants to hear from us.
Great. Next question is from Marsha Jimenez. So are the slides going to be shared afterwards? This information is super useful for future grant proposals. Yes, it is. So I could tell you that I'm quite sure he'll be willing to share, but let's have Sid answer that. Yes. And also this is being recorded and we make the recording available to the entire scientific community. So if you know of anyone that wasn't able to join today, we usually send out the recording link. And so, you know, feel free to share that information with your internal networks. So our next question is from Titli Muherji. I'm sorry if I did not pronounce your last name correctly. Was there any differences observed in the amyloid and tau PET scans among the diverse groups?
Does anybody want, I mean, so that wasn't presented here. However, Dr. Karen Meeker and Dr. Meredith Brasky are looking very in-depth at this. And the answer is the whole ATN framework varies by race and ethnicity. And Dr. Annie Cohen is working on a publication for that. So the factors that contribute to amyloid tau neurodegeneration are different. The link between amyloid tau neurodegeneration and outcomes are different. And so I actually emailed Damali, and the title of the email was "We Were Right," I believe. When we first got the analyses, we're so just amazed to see this. And even the link between things like sociocultural factors and these brain outcomes vary. So the data is of so much importance. Please download it and run with it. Yes, we are seeing those things in their publications in PrEP. That doesn't mean you can't download it and look at it and publish it and do it in a novel way. So please do.
Great. Okay, I think this question might be for you, Ali. What do you surmise is the source of resilience?
Yeah, that's a great question. And if you took a look at that low resource, low distress group, I was calling them low resource, but really there seem to be kind of higher levels of social support. So I've talked a lot with the team about this, and I think we really need to have a better understanding of like family systems and structure and network. Those need to get integrated into the next generation of these analyses. And not only do we need to move beyond just looking at individual level variables, self-report measures, but think about community level variables too that could serve as buffers.
Great, thank you. This next question is from Michael Benania. I'm going to warn you, he's a PO in the end, so. So did you run analysis of the 3 resources distress groups broken down into 3 genetic ancestry groups to see if they're affected similarly or not, and or broken down into ApoE variant groups?
So I did look at positivity rates of E4 across the groups, and I don't think we saw any differences. I'll have to go look at table 1 of that figure of that paper again to confirm that. And we haven't looked at genetic ancestry. This was all based on self-described or identified race and ethnicity. I think that there's always room to kind of improve and think about other ways to think about racial ethnic groups and ancestry. I think that this is a great starting place and an important place, and that self-identified race and ethnicity has a lot of purpose in our research investigations, and we really need to also know that that's valuable.
Thank you. Next question is from Philip Adeneni. Is the long-term goal of HABS-HD to incorporate pathological brain samples from participants?
Yes, that's actually part of the renewal. We in the first U19, we're actually launching the lumbar puncture sub-study, which is going and so at the next iteration, we are planning on adding a brain bank as well.
Great. Next question from Lasia Serpada. So approximately how many participants have DNA methylation data? Is this available to researchers via application?
Is that me?
Yeah, could be any. I know Raul also is pretty well versed.
Go ahead, Raul, you get it. You're on mute. You're on mute.
I really don't have the number at the top of my head, but I don't think it's that many right now.
Okay. But I could tell you, just keep checking in. As the data becomes available, they will be able to share that data with you if you're interested. So
we update the database normally twice a year. We have a November data release coming that will be much larger. And so anything in the database is yours to use.
Okay. Next question is from Thori Kroll. Sorry if I mispronounce your name. How are RF importance score determined?
Okay, now I'm gonna need to tell Dr. Ryan to answer. I work with Dr. Fan Sang, that's our biostatistician, and I'm not an expert in random forests to explain that, so I'm sure Dr. Ryan can explain that better.
So random forest is a decision tree based analysis, and what you get is a RF plot that gives you the relative importance, and the RF score is basically a summary of the importance of those things. And so you can even create cut scores with the RF score, et cetera. If you'd like to get really in detail about that, Dr. Fan Zhang, who works with us, actually invents new AI/ML methods. He was on an NIA supplement for that. And so I'm happy to get you in touch with him. He loves talking about these things and can get as detailed as you want.
Great. And next question is from Carrie Gleason. Such impressive work. Congratulations, Sid et al. Are you using a population-based sampling strategy for recruitment?
Hey, Carrie, good to hear from you. So we're using community-based participatory research approach. Okay, so we're not doing random digit dialing, et cetera, because most of the literature would say that it won't work in this sort of scenario. What we do, however, do is we have very good understanding of population density by zip code, by et cetera, in our communities. And so we target those communities and we blanket those communities. And so it's kind of a hybrid approach, but it is a community-based engaged approach. And so, but we do track all of these things. We have over time, we got to do it again, but we will also do analyses of the cohort relative to the population of Texas, the population at large. Our prior work has showed that, the sampling strategy actually represents the community incredibly well. We have to do it with a larger window, but it is a community-based approach, Karen.
Great. Next question from Hong Lei. Wonderful presentations, and there's a lot of info for us to digest. Please send us the slides as promised. Yes, it will be shared. I guarantee you that. This is Sid. He will share it with you. Next question is from Catherine Crowley. Hello from Ireland. Yes, we do have a lot of international people who join. I make art and write poetry in response to data. You said that 66% of participants are non-white. I wonder how many people in total are involved. My interest is in the sociology of health and brain health worldwide. Also, are you exploring the arts in Texas and beyond ASA prevention measure way of looking at symptoms?
So we currently have we're a little over 4,000 people in the study. Like I said, we replaced for attrition, and so even when the total active cohort of 4,500 will be enrolled, there will still be more people in the study. By the way, I love what you're doing. I'm a neuropsychologist, and art has such a wonderful benefit for rehabilitation, and even with those with active and even late stage dementia. So I'm super supportive and love what you're doing. We aren't doing that yet in the study. I will tell you, I'd love to. The problem, and I'm always the one to blame for that, because I'm the one standing between the scientists and the community because of the respondent burden. People spend approximately 10 to 15 hours with us to do this study. And so there's a huge, what can we do? What can't we do? Now, one thing I will tell you is that adaptability, flexibility, we change our scales over visits. We have a core battery, but if someone, if we want to add this piece in this visit and this piece in this visit, we will do those things. That's the balance that I'm always responsible for, though, is, and like I say, I always take that blame when someone has a brilliant idea and I'm like, not yet. I'm sorry. I love it. But there is a workaround. So if you have an idea and you've got something that you think's advantageous, the clinical core reviews it. We have a list of things and we do rotate those things over time. And I truly love what you're doing.
Great, thank you. Next question from Claire Murphy. You showed the lower incidence of ApoE4 in Mexican Americans. What is the incidence of ApoE2 and 3?
That's a very.
Go ahead, Mohammed.
I was going to say that's a very good question. I'm actually out of town, so I don't have the data with me. But Dr. O'Brien, you may have it handy.
Yeah, so we have shown that back in 2013, we published about the ApoE4 frequency being lower. Hector Gonzalez found that among Mexican Americans, it was also lower compared to other Hispanics. Interestingly enough, ApoE2 is also lower. And so you see a preponderance of ApoE3, not 2 or 4. And I know Mohammed's looking into this. Our preliminary data suggests that ApoE2 is not protective either. So it's definitely ApoE4, ApoE in general is different.
Great. So Mohammed, we're looking forward to seeing what else emerges from your work.
Absolutely.
Okay. Few more questions because we've got 2 minutes. So I apologize if this was already asked or discussed, but what are the future considerations to test p-tau 181 versus 217 versus 231 in these communities? I remember GFAP was mentioned, but what about NFL?
So we have assayed NFL, we have assayed GFAP. It's just not yet in the database, so we have run it. We also have run p-tau 181. That actually is in the database. There's a grant right now under review that, if funded, will add the 217, 231, MTBR, and others as well. So absolutely, we have some of these, and we're going to add the emerging as well.
Great. One last question, and this one is going to be very important. And again, we'll send your questions if we didn't get a chance to get to them. So someone wants to know how to access the data. How do they email you? Is there an application process? Can you talk a little bit more about that?
Yeah, if you just type HSC Institute for Translational Research. You'll find it. Click on the web page. There's going to be an icon at the top that says request data. Click on it. Go in there, fill out your application, sign a DUA. I'll review it. It gets back to you. The slides that you will have has the link in it that you can get to it. So all you have to do but please fill out the form. I get people that don't, and then I have to push it back. But once it's filled out, you're going to click boxes. Those boxes are going to tell you what elements of the database are going to be in your database. So your database is cut for you. And then you'll get an email that has a link that tells you where to go download your database. It is very straightforward. If you're a student, please have a mentor so that we know we have someone there to help guide you. If you don't have a mentor, we will help find you one.
Great. Okay, well, we're at the 2:15 mark, and so we'll stop here. Again, I apologize if we didn't get a chance to ask your question, but I guarantee you it will be answered. We'll send those forward. So once again, thank you so much, Sid, Ali, Raul, Mohammed, for the great presentations and for all the great work that you and your team do. So thank you, everyone. Have a wonderful day. And
Thank you.
we'll see you guys again soon. Take care.
Thank you.
So our next webinar, thanks, will be speakers will be Laura Zahudi and Christine Aruch, will be speaking on their work with the Arab American population, and another great research group. So please feel free to join us September 2024. Thank you. Take care, everyone.
Thank you, everybody.
Bye.